back to Holistic Health Masterclass Podcast. This is Brett, and I am coming to you here on March 18th. We are mid-pandemic, and things are shutting down real fast all over the world. So wherever you are, I just hope that you're doing okay, that you're staying safe, that you are self-isolating, and that you're taking the necessary precautions to protect yourself and your family. I don't want to make this a somber episode because that's not really what it's about. And I also don't want to make it a long episode because my recording that I did, which I'll get to in a second, is long enough on its own. Okay, so I'm just going to leave it at that. I'm going to be, depending on when you're listening to this, I'm going to be doing a distilled podcast on its own that sort of discusses a few of these things that I'm going to talk about with Dr. James Lyons-Weiler today. I want to talk about them in more simplistic terms, okay? Because I understand that a lot of you listening to this are maybe not scientists or not health professionals. You don't understand the lingo, but I wanted to leave this recording in its entirety as it is, okay? It saves me time on editing, but also for those of you that are perhaps more science-minded or doctors or medical professionals and so on, you will appreciate James going into some more detail on some of the finer points and the nitty-gritty. I also just want to point out that, there are things that we know, there are things that we don't know, there are things that we think we know, and there's things that we just know absolutely nothing about at all. So just bear this in mind and put this in context when you're listening to this. Things are moving real fast here, and there are constantly new developments happening. So I've done my best to try and not get too far ahead of the story and to give you the best snapshot of where we're at right now. All right, so my guest today is Dr. James Lyons-Weiler, also known as Dr. Jack. And for longtime listeners of the show, you would have remembered him from a previous podcast that we did on vaccines. I'm not going to get into his whole history and his whole bio. Suffice to say that he is a PhD scientist. He has designed many studies. He's worked with some of the top universities in the world. He's an esteemed and respected scientist and is also very, very good with predictive modeling. You'll get a sense of that when you listen to this podcast. But here's the snapshot of what we talk about, right? So we talk about some of the basic stuff. So how is this transmitted? What are these sort of incubation periods? What should people do? We talk about self-isolating and self-quarantining and why that's important. We obviously talk about the spread, right? And I ask a very blunt question, should we be concerned? Is this pandemic actually going to blow up? So we talk about that and we get into the ins and outs of that. We also talk a little bit about the whether or not this is man-made, because this is something that's going on out there. People are suggesting that this is man-made, that it's 100% bioweapon, that it's manufactured in a lab and genetically altered, etc., etc. And I've now had multiple reports. There's a study that was published in Nature as well, the journal Nature, which shows that this is not in fact man-made. Okay, so for those of you that are perhaps a little bit more conspiratorial, I'm just going to leave that subject right there, and I'll let you debate that. Hit me up online as well if you want. We spend a good deal of time talking about what we can actually do, right? So we do talk about nutraceuticals and some novel supplementation that may work. I'm going to be doing a separate podcast on that. So if you're listening to this in the future, there will be a link in the show notes. If you're listening to this fresh right now, today, March 18th, you can expect that podcast within the next couple of days, okay? So we talk about nutraceuticals, what supplements actually work to the best of our knowledge based with current science. And of course, we talk about vaccines, right? So, we're looking now at everyone is scrambling to find a vaccine. And miraculously, we have the Chinese, we've got the Israelis, we've got the Canadians, we've got all these different countries around the world that miraculously 2.5 months post-discovery of this virus, we all of a sudden have a vaccine that is ready to go with human trials. At time of recording, they are in fact doing human trials in Seattle, I believe, and they've mustered up 45 participants with no placebo controls. And of course, as you will hear hear in this podcast, there are genuine concerns with regards to no animal safety studies, and also looking at the previous SARS vaccine and what happened with those animal studies there as well. We talk a lot about reinfection, and this is an important point, right? You see in the UK right now, they've actually pulled a full 180. So their approach was going to be let everyone get infected, and thereby we would create herd immunity. And they really just did a full 180 on that because they realized that the implications would be extremely serious. Part of the reason why that would be serious is we don't really know how this affects people yet. Okay, there are more than likely underlying genetic factors. There are also comorbidity factors, so people with pre-existing cardiovascular disease, diabetes, metabolic syndrome, cerebrovascular disease, and so forth. But when you talk about reinfection, it's starting to become a little bit clear that people who have either previously been infected with SARS, so the original SARS, or MERS, so MERS, Middle Eastern Respiratory Syndrome virus, so that's MERS, it's now starting to look like these folks who've been exposed to those viruses previously have a much more aggressive response to COVID-19. We obviously bring into the fold the vaccine, and we talk a little bit about why and how this vaccine might play out. And quite frankly, that the vaccine is not necessarily the answer, and it is not going to save this pandemic at all. So I think that you're going to find this quite interesting. There's a lot of really, really useful information. And again, for those of you that are not super sciencey, I'm going to be putting a link to a shorter summary where I'm going to speak about this in very plain English. Right, so I have a lot more to say, but you're going to be hearing from me through Facebook and elsewhere. I want to talk about supplements. I want to talk about what you can do and what's going on out there. So just stay tuned to the podcast. If you're not following me on Facebook, just check out the links on the website. And anyway, so this recording was actually a Facebook Live that we streamed a couple of days ago. It's up on YouTube if you want to actually watch the video. So check out the links in the show notes there. You can see us talking if you want to look at some bobbing heads. And so that you'll just hear that in the introduction of this episode. All right, so I'm going to leave it at that. Thanks for tuning in. And here is Dr. James Lyons-Weiler. All right. Hello, everyone. We have obviously had some tech issues, so we are coming to you live through Facebook, multiple platforms. And hopefully those of you who are following me, Brett Hawes, you are on James's platform. But either way, whether you're catching this live, whether you're catching the replay, we are here with some very important information and a bit of an update and a bit of an expansion on what's going on with the coronavirus pandemic and so forth. James, I'm joined today by Dr. James Lyons-Weiler. James, thank you for joining me today. How are you?
I'm well, thank you. And thanks for interviewing me. This is an important time. Thanks so much.
It is indeed. And I think, there's a lot of panic. There's a lot of chaos. There's a bit of confusion. And I think there's also some things that we are being told and things that maybe we're not being told. So I just want to sort of preface this entire discussion today. For those of you who are not familiar with me, first of all, I will introduce myself. My name is Brett Hawes. I am a functional medicine practitioner, clinical nutritionist, and a professional educator. I host the podcast Holistic Health Masterclass podcast, and that is partly why we're here today. I've actually had Dr. Jack on my podcast before, and we literally conspired, I would say a little bit later last night to hop on this call this morning just to unpack some breaking news. So the purpose of today's live session is twofold, probably threefold. First of all, I want to sort of cut through some of the noise out there because the media has created a lot of panic, a lot of hype. And what I want to do today is I really want to try and decipher and discern what is hype and what is actually in fact real. So there's that side of things. The other thing I want to look at is, and acknowledge is that there's things that we know right now there's things that we suspect and we can speculate, and there's things that we absolutely don't know. So at time of recording, if you're watching this even 2 or 3 days later, because this is such a fast-moving story, some of the information that we share here might have changed, might in fact be incorrect, but we're going to do our best here at this particular time, March 14th, at 9 a.m. or 9:15 a.m. We're going to do our best to pull some of this together for you. So Dr. Jack, and I'm just going to call you Jack here. Mm-hmm. Now, I was trying to figure out a good point to start our discussion today, and I think what I would like to do is just hit some of the easy points, right? I want to avoid rehashing what media is saying and what most people know. So obviously, when we look at the numbers, this pandemic is in fact exploding. There's exponential growth in all countries, for that matter. But just this morning, we woke up to reports that the EU is now the epicenter for what's actually happening. So first and foremost, very, very straightforward here. How do people actually catch the coronavirus? Because, there's reports of it lives on surfaces for X amount of time, it can become aerosolized, et cetera, et cetera. So let's just start there and we can sort of move our way forward.
Sure. Thanks, Brett. So great. So opening question. Clearly, the initial reports that this virus has an unusual and uncharacteristic for coronavirus asymptomatic period. Those reports are correct. It's the only thing that could explain the rapid spread of the virus. I've done modeling. Other people have done modeling, and we absolutely have to have an asymptomatic period to see the number of cases. Obviously, touching anything in public that has a virus on it, or shaking hands or hugging or kissing and that kind of thing, is an easy way to transfer almost any virus. But this one also apparently can be transferred through the air. I'm calling it partially aerosolized, but it seems like people that were in buildings and on cruise ships that had closed circulation systems. There's one report from China where a person was tracked. He had no contact whatsoever with anyone who had a diagnosis when they were doing contact tracing. And they found him on closed-circuit television entering a bus, and the person that had been infected had left the bus 4 hours previously. So it seems like it has a lingering tendency. It's highly infectious is the word, which is a different. It's highly transmissible. It's a very different idea than lethality, which is virulence.
So okay, so I mean, obviously the regular precautions, washing hands, avoiding contact, and so forth. But here's another thing, and again, I'm just trying to figure out how to unpack this for people so that it's understandable. Why have we seen such a rapid escalation over the last literally 10 to 14 days? Because you were actually sounding the alarm probably 2 or 3 weeks ago now, and really saying we need to constrain this, we need to limit contact with one another and so forth. So how is it that, whether it's the authorities or why are we just so late to the game, and why is this escalating at such a rapid pace?
That's a great question. So there are multiple social factors that have been at play. Among those social factors include, let's back up a little. We had clear data from China and other countries where the outbreak had gotten out of hand. And we also had data coming in from Italy. We knew some of the characteristics of this outbreak, and including the clinical symptoms. And people say, well, how do we know? We'll get to testing, I'm sure, but how do we know that it's actually coronavirus and not some diagnostic substitution, and that they're trying to control society, and that this is some kind of a hammer that's being slammed down? It would take an extraordinary amount of planning for this to have been planned way back in 2005, and I'll get to why I think that that's unlikely. But nevertheless, the CDC distributed some tests, shipped out some tests to the Princess cruise ship. and those tests had a tendency for false negatives. I had done an analysis of the constituent parts of the test that make the test work, and sent the results to CDC. And now I have alerted the FDA, and the FDA has responded with great interest about the specific mechanical things that are wrong, or biochemical things that are wrong with the test. So obviously, if you have a coronavirus and you're asymptomatic and you test negative and you're let go from the cruise ship, you can spread it that way. I don't want to do finger pointing, but the presidential administration clearly has favored business as usual in terms of the economy for as long as possible. They needed a window of time, I think, to get their particular ducks in a row. But in reality, this virus has been traveling to the United States at least, probably since late November, early January. And we're talking about hundreds of thousands, if not millions of people then who had it. And this may explain, it must have been here much earlier than the cruise ship, because this may explain why we hit the hockey stick part of the curve so fast. In my modeling, if we were complacent for 18 days, it still took, months, a month or two, before we would reach the hockey stick with the characteristics of this with this virus. So, we're looking at a situation where it's already entrenched in the United States, and people are waking up to it because it's starting to reach those parts of the population that have the most serious illness from it. And so I think it's all these factors. It was here far earlier than we thought. There was no response. There was an inadequate response to the early calls to at least tell people to stop hugging and kissing. You're right, 2 or 3 weeks ago. Stop putting your hands above your shoulders, that kind of thing. And now the social distancing that the voluntary quarantine that's been suggested, I think will morph into mandatory quarantine. And people need to understand how important that is from the perspective, I'm the last guy to roll over to totalitarianism. But people have to understand, if you enjoy the comfort of your home, if you're under mandatory quarantine, don't break it, because you will be away from your children, your house, your food, everything. You'll be away from everything that you know and love, because they'll put you into a mandatory quarantine situation. If you break a mandatory quarantine. As far as I know, there's no mandatory quarantine so far, but I haven't read all the headlines this morning.
Right. And as I said, this is such a breaking story that we're starting to see. I mean, even here in Canada, they're talking about literally sealing up the border. I might be slightly incorrect with this, but I believe that cruise ships are no longer allowed to dock in Canada. So that's the closest or soonest docking date will be July, I believe. So, yeah, this is moving at breakneck speed, and borders are getting sealed up real tight. But let's hit another question that's probably on everyone's mind, and that is, there's a lot of memes floating around out there, and one of them is, this is just the regular flu. We don't need to panic. It's just the same. It's a mild cold. And I want to talk about the first thing is obviously, is that correct? What are we looking at in terms of rates of infection, and then to follow that, rates of mortality? And then the second one is, we're obviously starting to see different populations are being affected differently by the virus itself, right? So, I think something you posted late last night, which was a research study showing that people in Wuhan and Hubei province are way more susceptible, and the symptoms that they're experiencing are way more aggressive than, say, people here in the West. So maybe you can sort of unpack that a little bit for us.
Sure. So there's a couple of questions there. The rate of reproduction of the virus is not an intrinsic characteristic of the virus. It depends on how the host species responds. And since we have culture and intelligence, I'd like to think we have intelligence, culture and intelligence and technology, we can actually modify the rate of spread, as everyone clearly understands now, I think, by changing our behavior, by not aggregating into large groups, by not touching our face, by not, these kinds of things. But they are not, there's estimates that range, it's above 2 to maybe as high as 4 maybe a little bit above 4. And what that means, the R naught, is the reproductive number. That's a basic reproductive number. Now, some people tend to call it a rate. It is not a rate, because a rate measure requires the number of infections per unit time. It is simply a number. During an outbreak, it is understood that a person will infect 1, 2, 3, or 4. Okay, so the entire rate is that until you're no longer capable of transmitting. Okay, so don't call it a rate. It's R naught. It's the number of people that you're likely to infect. And, the cool thing about simulations and modeling is that we can assume that we do nothing, and we just take a lackadaisical approach. We can assume and modify the amount of social distancing, and we can model in, and I did model in, the effects of therapeutics and treatment. And so the mortality rate is linearly related to your to age. So if you have anybody who's it's not unique to the elderly, but if you have anybody that's elderly and has other conditions, health risk conditions, they are at most risk, and they should be protected. And don't go visit grandma, leave groceries on the doorstep and things like that. So that there's a 10% case fatality rate in people that have a cardiovascular disease. There's a 6% case fatality rate in people that have diabetes. Well, look at the United States of America. What do we have? 45% people that have metabolic syndrome. And so we're a bit of a sitting duck here. Certainly, there's been a lot of discussion about the biochemical molecule ACE2, which is a receptor on lung epithelial tissue that the virus enters into the cells by. ACE2 is very well known now by a lot of people in our community to be increased in people who smoke. And so if you smoke, if you know someone who's smoked, now's the time to take the opportunity to finally kick the habit, for sure. If you have multiple risk factors, you're elderly, you smoke, you've got diabetes, and you've got, chronic heart condition, a cardiovascular condition. You're on high blood pressure medicines. You want to be sure that you do not get this virus. You want to be sure of that. Now, the difference that you asked about in Hubei province, was something that I predicted could and was very likely to happen. Specifically, when I first made the phylogenetic analysis about where this thing came from a little bit, which is another completely important discussion, but we're kind of going to stay clear of that for this. What are the facts that people need to know now?
Exactly.
That in Hubei province, I suspected that there had been a phase 2 or phase 3 vaccine trial that the Chinese didn't tell the public about or tell the world about, so they could get a jump on the market of a coronavirus vaccine. And the reason why I suspected that is because we know that the Chinese created, for SARS, they created not SARS-CoV-2, but for SARS, they held a phase 1 clinical trial. And they had something like 120 people that were registered in that clinical trial. And then the next step would be to assess safety, to take a look at larger populations. Now, this clinical trial happened a long time ago. It happened like 2008, 2009. And there was kind of a worldwide moratorium over doing human vaccine safety studies with SARS, not because of the anti-vaxxers, because in 2008 to 2016, 2015, where was the vaccine risk aware public that now exists? It was a much, much, much smaller movement. But in reality, it was because the animal safety studies that were done actually showed high morbidity and mortality in mice and in ferrets that were injected with the SARS spike protein as a vaccine, and then when the mice were infected with the virus, then they had a very serious mortality rate. So that's the reason why why we don't have a SARS vaccine. And Peter Hotez was testifying to Congress earlier this week, and he completely misled the American public. He completely misled that committee by saying that it's the anti-vaxxers are the reason why we don't have SARS because we couldn't get funding. He's not going to get away with it either. We're going to get into this in deep, but he's not going to get away with it because everybody that's watching this video, I predict by the end of the video, will be calling your congressmen and senators. So you might want to start looking up their contact information.
Right, and we'll definitely circle back to that. I think just to sort of boil things down for people, are you, and just to sort of throw a couple of general questions that maybe we can answer in a very straightforward fashion, should we be very concerned based on, like, from my understanding, the R naught, or how many people each person would infect here in North America anyway, is actually quite low as far as I know. Like, I've heard other scientists and doctors from Johns Hopkins, for example, that have said they're not really concerned about that R naught. And I don't know what that number is. Perhaps whether it's one person infects 2.4 or 2.8 people, somewhere around there. But the other thing is, the exponential spread, right? I mean, are you, as someone who does predictive modeling and translational science, like, are you concerned that this is really going to blow up?
Well, we're actually seeing that it is blowing up, so there's no doubt about it. I think that doctor will be rethinking his position. He may not be concerned about the R naught for the general population, but he certainly should be concerned that patients will be walking in that have, for any reason, like a broken arm, a twisted ankle, and carry the coronavirus into his medical facility. We need to understand that the virus doesn't actually wait for you to have symptoms to infect the doctor that's examining you. Our medical frontliners are going to be swamped. There's no doubt about it. Let's break down the expected. Let me answer the first question. The actual R naught in the United States will change from week to week as the steps to socially isolate, to get this under control. What we want to do is we want to change the inflection of the curve from exponential, which is where it is now, to flatten it out and then turn it down. And the way to do that is to target this R naught. We have a situation where there's been this virus that's been circulating. I just saw a report that the estimate there's 100,000 people in the state of Ohio alone that probably have it but don't know it.
I saw that. Yeah.
And so if that's true, and each one of them, infects more than two people, because as they go, your kids go to school, you go to work, you're shopping, China was burning currency because they figured out that the virus could be passed on through dollar bills, their currency, paper currency. I'm going to go with the safe assumption and say that this is spreading at a rate that's greater than 2.1 in the United States. Okay. And the reason why I say that is the shape of the curve tells us that we can expect at least a doubling of new recognized cases. At the point of inflection in that curve, when it's at its maximum, people think that it's an exponential curve. It's not. It's actually a sigmoidal curve. And I hate to get technical, but what that means is it starts out flat and then it goes into a point of inflection where it's basically taking us straight up. And that happened so quickly. For people to say this is just like the flu, no, it's not, because the flu over the same time period is going linear increase. Okay, it's much lower in terms of the rate of new patients. So if we've reached this point of inflection, then what happens in diseases in a population that are infectious, they will start running out of new people to infect, and then the curve will flatten out. Right. That's a sigmoidal curve, and that's where China is right now. China is, as everyone knows, what we see is draconian measures. We see the people, being arrested, being thrown into ambulances, and this kind of thing. We tend to think that that's a general thing that's happening, and it's not. What my understanding is that, and I'm not making an apology for the Chinese government for handling people this way, but my understanding is that these are people who have multiple times broke quarantine, or were trying to travel out of the province after they closed a border. And so you can expect, unfortunately, I hope it doesn't come to that, but I think we can expect, because people will tend to act selfishly, There was a medical student that was traveling in Europe, and he came back home and he didn't self-isolate, and yet he knew that he had been someplace where there were people with this infection. And he came home instead of self-isolating. He went to the mall. He went out, got something to eat. That's a very selfish act, because he may have offset a new chain of transmission that could infect millions. It's very, very important that you understand that we are part of the problem, and it is very difficult for the human mind to accept. There's one person I know that I've been in contact with about this since January, and this person hadn't seen a neighbor in a long time, and they just a few days ago they saw the neighbor and they went over and gave him a big hug. And I was like, caught you caring. You got to. It's not that they don't care. It's that that definite kind of gut react, the gut awareness. Intellectually, we know the math. We know that we should isolate.
Yeah.
The gut reaction is, but I don't want my kid to be stuck in a quarantine at his friend's house, so I have to break quarantine. If you break quarantine to go get your kid at a friend's house rather than call the authorities and say, I would like to transport my child, how do I do that? Don't think that there's not, even if you see your neighbor, in a stressful situation, the first thing to do is to say, okay, how do I do this? I need my child home. I don't want him over there. So I'm not asking people to bow to the state. I'm asking people to use the state. Don't give up your rights. People don't give up. The first place that people give up the rights is in their heart, where they give up and they say, okay, the government's out of control and we can't do anything about it. Don't give up in your heart that there's a human being at the other end of the phone when you call the health department, or who, by the way, is most likely working from home. They probably are setting up phone lines for people to answer these calls from home. So they too are isolated. We're really all in this together.
Yeah. Well, and I think, again, it's just smart, sort of sensible advice. So another big question, you brought it up earlier, and I think it's probably a good thing to discuss is testing. Okay. I mean, that's been making a lot of news headlines lately. Access to testing, is the testing accurate? How many people out there are actually, infected and do not have not been tested? So I don't know where you want to start with that, but maybe you can give us some numbers and how this is all shaken out.
I want to start with the history books will blame the rate of spread in the United States on the CDC. The World Health Organization, had a viable test. It was made in Germany, German engineered test. The CDC said, no, thank you. This was a few weeks ago, where we could have been having testing, and everybody understands the importance of accurate diagnostics from this setting now. And they said, no, thank you. We have our own. Ironically, that is the exact same thing that people at the CDC in their diagnostics said to me when I had created the Ebola Rapid Assay Development Consortium. They were going to do blood draws in the airport to do PCR testing for Ebola during the 2014 Ebola outbreak. And I had realized that was going to be a problem, so I pulled together 3 universities under the Ebola Rapid Assay Development Consortium, and we were going to use a colorimetric test that wicks your saliva into a bottle, and then you drop the bottle into a plastic bag and seal it, and then you hand it to the person, so you're not bleeding all over the airport. And so this testing could have been started 3 weeks ago, and the mysterious number, that denominator of people who were tested, only matters for a short period of time now, because pretty soon everyone will be infected, is the expectation. Unless they get a test out there, and then they can really triage people. Medically, they can triage people. So the test had some technical flaws that I mentioned earlier. I communicated those to the CDC and the FDA. They should probably ignore part of the test. There's, I think, 4 of the probe sets that will amplify human protein encoding genes. They should just ignore that part of the kit and use the rest of it. It shouldn't lead to a delay in the rollout of the test. They should just have a different interpretation. They use a 3 out of 4 probe set criteria. They should really use a 1 out of 2 or 2 out of 3. And ignore the ones that they have are likely to give you a false negative. And so we really don't know, say, an actual number. But again, if you look at the curve, it really super matters at this point in time, when the infection is taking off. If you be able to triage, it matters here too. But at the point in time when you have so many people that are sick and the medical community is already overwhelmed, testing's not going to matter anymore. It's really not going to matter. That's why they've moved to presume positive, where you say, okay, listen, it's better safe than sorry. Go home and quarantine. You're one person. We recommend that you stay home for 14 days. Sorry about the inconvenience. We don't know your status, but all right. And at that point in time, they're not going to be talking to individuals. They're going to be saying, okay, well, if it's good for them, it's good for everybody else. With 100,000 cases, how can you overnight determine which 100,000 cases in Ohio have the coronavirus and which don't, even if you could test everyone?
Right.
It's not tractable.
Right.
It's just not a feasible operation.
Okay, so that's good to know. Do you feel that the numbers that are being sort of thrown around are that we suspect that at maybe the height of this epidemic, that 60% of the population will be infected? Do you think that's a good number, or higher or lower?
I think that determines on what happens. Should have started 2 weeks ago, but we still have time to reduce that percentage of infection. And, we can see at least 50%. Now that I know that they're shutting down schools, now that we know that there are people that have kids and have to stop work and go home to take care of their kids. The problem with shutting down schools, of course, is that about 20% of the nurses' workforce in the country have school-aged children. And, there hasn't been a lot of kind of forethought. And this is what I was saying last week. If we're going to shut down the schools, make sure that you have some kind of a daycare sharing situation for the kids of the nurses, for the kids of the doctors. You have to take care of these people, not that they're a separate caste, but you have to make sure that they're able to do their job. Because with 6% of people with this infection, getting the ICU, requiring the ICU let's call it up to 66%, because there's something strange in the Hubei province that we talked about. And 14% of people, both medical and non-medical, just regular people, having serious illness. I think we have something like 750,000 beds in the United States. We really need millions. We may have 46,000 ICUs beds, and we're going to need, hundreds of thousands, hopefully, and not millions of those. The problem is that the medical community becomes overwhelmed, and they can't handle other things that they handle. And so there are other people that have conditions that are equally lethal, and those other conditions can actually become neglected, and people can be turned away for a hospital bed because they're so overwhelmed about whether or not this is going to be contagious to their staff and to everybody that works in a hospital. But if we could bounce back to Hubei, this question made me remember that there's a bias towards men in terms of being seriously ill, so far as we know from the Chinese data. But the Chinese have a very, very huge gender bias for smoking. So it seems like there's a number of factors that could be going on here. I'm not saying that they did do a phase 3 clinical trial, but I wouldn't be surprised if they were trying to get a jump on it. I don't have any independent data. I've tried to look for it that shows that they did a phase 2 or 3 clinical trial in Hubei that sensitized people. But that's really important to think about the sensitization of being exposed to that spike protein.
Well, okay, so I want to come back to that in just a second, because I want to address the elephant in the room, which is obviously the vaccine as well. So we'll get to that in a minute. So are we seeing that the mortality rates, I've heard the number 0.6% as the sort of mortality rate. Is that an accurate number?
Well, that 0.6% overall, you're talking about in the United States or worldwide?
Worldwide. Obviously, it's going to differ slightly from region to region, which we'll get into in a second.
Right.
And through different demographics of population as well, obviously.
Yeah,
right.
Sorry, I'm going back and forth. So when people say, well, maybe we don't have so much to worry about, because certainly a 25-year-old person probably doesn't have much to worry about. I'm 50-something, and so there are 35-year-olds who have died from this. And if you're trying to assess individual risk from 0.6%, it's just like the flu. It's a bad mistake to make, because the flu doesn't have as quick a rate of spread of so many people getting sick and dying at the same time. And so what we're trying to do is we're trying to do social distancing and isolation, and now I see that they're funding therapeutics as well. I just saw that some philanthropists, including Bill Gates, probably got the IPAC memo to go ahead and start funding therapeutics, because social distancing alone won't solve this. We actually need antivirals on the table. We need vitamin C. We need, herbal therapeutics, anything that will slow down the virulence, reduce the virulence of the virus, reduce the rate of transmission of the virus. When people say, well, we don't have to worry because it's 0.6% worldwide, I point to Italy. All right. Italy is a European country. Italy has Mediterranean lifestyle. They drink red wine. They eat the Mediterranean diet, so on and so on. There's something about Italy. Or I point to Iran. Okay, there's something different about Iran. And I already said earlier, 45% of Americans have a precondition that predisposes them to serious risk of serious illness, critical illness, and mortality from this virus. We are not a random group of people from the rest of the world. Most medical doctors who've looked at this said that, yes, we could be worse off than Italy as a country.
Oh, wow. Okay. So, and that's good to know. It's important to know. You brought it up. Let's talk about therapeutics. Obviously, the narrative that I keep hearing out there is everyone loves to talk about numbers. We love to talk about how, what could potentially happen, how bad things are, anecdotal stories, et cetera, et cetera. But when it comes to the topic of what can we do aside from washing hands, social distancing, isolating, quarantining, et cetera, there's not a whole lot of talk about actual therapeutics. And then most people are defaulting to, until the vaccine arrives, keep washing your hands, quarantining yourself, etc. So let's leave the vaccine topic alone just for a few minutes. And are there therapeutics that are being implemented on a large scale? Whether, vitamin C, I think that's been thrown around a lot. And correct me if I'm wrong, I do believe that they are actually curing people in China with vitamin C, whether that's intravenous, whether it's orally taken. But let's start with the therapeutics. What have you heard? Do we have antivirals? Do we have access to these things? And do they work?
Well, that's a great question. So I'll direct people to jameslyonsweiler.com. There's an article of 21 key points that you need to know about this coronavirus. It includes the studies, the science that shows which antivirals might be useful. One of them is chloroquine phosphate. The Chinese have a study. They are using it, and they're starting to get things under control there with a very, very few new number of cases per day now. High-dose vitamin C, there are anecdotes on that. The thing I want to emphasize that I think that we should really explore very rapidly in the United States is to get the people who have survived the infection, that have antibodies, right, into a triage situation where they can find out who can donate and who cannot donate plasma. And you take 1.5 liters if you're an adult of plasma. It's a lot, but we need it. And you isolate the antibodies and you create a product that's called convalescent plasma therapy for convalescent plasma therapy. The survivors, because there's going to be hundreds of thousands of survivors who had mild illness, there should be plenty of convalescent serum around. The CDC doesn't want to do this because, they want to put on their superhero cape and they want to bring the vaccine to America. They think that they're at the end of the script of a movie and save the world by injecting a needle into their thigh and say, look, I did this for you, Dad. But the reality is, we're sitting on top of something that could really be, I think we must do it. And if people who are involved in seeking therapeutics go, the philanthropists, if they see this video, they go to jameslyonsweiler.com. There's 21 objective points. They're backed by science. But I don't think I emphasize convalescent plasma therapy enough because there are people with the cure in their bodies walking around. So how do you, if somebody's, I get messages all the time with questions, and I'm sorry if I can't keep up with everyone's questions. But people say, well, is it more dangerous? Is it as dangerous as, say, a vaccine? There's no adjuvant. There's no all the other stuff that people don't want in their vaccines. There's no aborted fetal cells and so on. What we have, unlike vaccines, what we have is a series of antibodies that, for frontliners, especially in the medical community, probably need to get this up front. And I do want to say that people are going to be calling and have called President Trump selfish because he hasn't been tested. I want people to remember President Trump is the not only the executive in chief of the United States of America, he's also the highest ranking official in the U.S. military. He's the commander in chief. So if you don't think that the military has already protected our boys and girls, okay, with convalescent serum plasma therapy, he is protected by some other means. I'm sure he's the president of the United States. It's if you're a liberal and you're on the side of it, he's like a punching bag. He's like a clown. But in reality, in every way, shape, and form, no matter who sits in that office, they are going to be given the ultimate protection because they are commander in chief of the military. So we really need to start. We need to stop politicizing this. I don't care what side, what party you're part of. I don't care if you're an independent. I don't care if you're a liberal. I don't care if you're conservative. Everyone needs to stop political talk. I'm talking about Nancy Pelosi. I'm talking about Republicans. It is absolutely beyond the pale at this point to politicize this any further. We absolutely all have to pull in the same direction because this virus doesn't check your voting card. Just like a vaccine doesn't check your voting card, or whether you're religious or not, if you're going to be injured by a vaccine, this virus doesn't care. It's the honey badger virus. Peak prosperity. I got to give a shout out to Peak Prosperity. That's their phrase. That guy's great. He's been on this every day. But chloroquine phosphate is important. Convalescent therapy is important. And things that you think are antiviral. And before the medical community goes crazy and says, wait a minute, you're going to give them some arbitrary herbal, you're encouraging people to try herbal supplements. You're crazy. This is not a crazy position because I'll tell you why. I'm going to show it with my hand again. This is what happens if you do nothing, right? To this, and it takes over the country, and we lose 10%, 20% of our elderly. This is what happens if you do just social isolation. It stabilizes. It actually oscillates like this back and forth. Okay. This is what happens if you just do if you just do therapies. It stabilizes at a high rate. But if you do a combination of social distancing and you reduce the virulence of this thing and cut it down with anything that works but doesn't hurt, anything that could help but doesn't hurt, then it goes up and it crashes. And that's what we want. Right. We want to get back to work. We want to get back to school. We want it to go up, stop, and nosedive down. And the simulations show this. And so if somebody has a tea leaf extract that they think is antiviral, if it's not going to hurt you, sure, take it. But don't count on that alone. Count on, if they offer you chloroquine phosphate, if they start distributing antivirals, if you're in a really high dense population area where people have to get out, or certainly if you're on the medical front lines, you absolutely start to, and this is what I want to say. One thing that I need people to ask their congressman for right now is a hearing on why the CDC has not brought forward convalescent plasma serum, plasma therapy. That's issue number one. Get a hold of your congressman and senator today, get their email, call their office as well, and say that you really absolutely want a hearing on why CDC has not mentioned convalescent plasma therapy for the United States citizens.
And, I think it's also important to point out that the Chinese are actually there are many, many clinical trials that are being done on herbal therapies right now over there. Their medical system is essentially half allopathic and half traditional Chinese medicine. So let's not, discount, because everyone is sort of waiting for the pharmaceutical solution to kick in and, the drugs and whatnot. But I think, if we go down to the health food store, we can shop online, we have access to antiviral herbs. Some of those would include and are not limited to things like olive leaf, berberine, golden seal, oregano oil, vitamin C we've spoken about as well. So there are many, many things that could potentially help. But I think what's also important to understand is that, because people love to shoot everyone down, where's the science, where's the literature? And while we have some literature and some science, I think it's also important to point out that we are dealing with something that's novel. So all of this stuff is happening and emerging right now. So best to just be prepared and do what you can. Again, as you said, do all of the other preventative measures to essentially give yourself the best fighting chance that you can.
Agreed. Now, let me say one last point on that.
Please.
Selenium is one I didn't mention. N-acetylcysteine, spirulina, and high-dose glucosamine. They're looking at remdesivir. We have studies that show that chloroquine and remdesivir inhibit the entry of the 2019 nCoV in vitro. So the studies are there. There's also an active compound in licorice roots. My point is, what would they have us do? Self-quarantine for, 3, 6, 9, 12, 24 months? Self-isolate for that long while they figure out their vaccine? Or would they also like to know that the population is trying to bring this thing to an end by using things that can't hurt them that might help? And
exactly.
This is like you said, we're breaking new ground here. I think it's very important that people not try anything that is sold as snake oil, and don't reinforce anybody that says I have a cure for, COVID-19. Ask them for the clinical study. If they don't have it, then look at the product yourself and say, okay, is this something that could potentially harm me? Does it make any sense that this could potentially harm me? And, be really careful about taking anything that you wouldn't otherwise be able to take. Like if somebody says, well, I have a new extract from, it's a mixture of gasoline and astragalus root. No, I'm not taking it. I'm not going to be putting gasoline in my stomach.
Yeah. Well, I think what's important to recognize is that there are tried and tested antivirals, generally speaking, general antivirals out there. You and I have both listed some of those right here just in these last few minutes. Okay, so let's address the elephant in the room, which is the vaccine. Everyone, as I said, is sort of hoping and praying that we're going to have a vaccine. And I just want to sort of paint the picture here and preamble a little bit. Typically, vaccines take many, many years to develop. Depending on the type of vaccine, that could be anywhere from 8 to 15 years. And coincidentally, we now have the Chinese who are saying that they're ready to go, probably in April. That's been thrown out there. Those are news articles. We now also have a Canadian company that says that they can manufacture a vaccine that is not cultured or grown in egg. It's actually, I believe it's plant-based. I'm not quite sure what that means exactly, but they can turn a vaccine around in 20 days. Now, where do we start with this, James? What do you have to say about all of this?
Well, let's start at the beginning. And let's say that I was to develop a vaccine. The first thing that I would have to do is, find some component of the pathogen, a bacteria or virus or other organisms that I think have potential use in the vaccine by being immunogenic. That means that they're capable of mounting an immune response. And once I've identified those, I have a choice of technologies that I might use. I might use a killed vaccine, the killed virus or bacterium. I might use isolated proteins. I might create a virus-like particle that has the proteins on the outside or the shell or the coat of the particle. Whatever I use, I then have to go through animal studies. The animal studies are there so that we can see if there's any unintended consequences of using this particular vaccine on mammals. So usually it's done in mice, mice or rats or ferrets. And the outcomes that are looked at include the efficacy of the vaccine. So you vaccinate an animal, and then you challenge the animal with the virus or with the pathogen to see how many develop symptoms. They already have antibodies, hopefully, from the vaccine. But you want to see if the vaccine is effective. And so you look at the efficacy this way. The second thing that you want to look at is whether or not there's any unintended consequences. And so you look at the health outcomes of the animals. So it's, you don't just look to see, okay, well, yeah, okay, I've got a vaccine against, say, the measles. I did the animal study. And by the way, I doubt that there was any animal studies, but that's a whole other thing for somebody's.
We'll get to that in just a minute.
Yeah.
Don't worry.
But for me, okay. So you inject the antigen source, and then you look and say, okay, do we see the disease? That's one thing. The other thing that you should look at is see if there's any unintended consequences like autoimmunity. What's the risk of morbidity or disease? What's the risk of mortality? Imagine doing a study for coronavirus and finding that all the animals that were vaccinated had a higher risk of, serious illness from the very virus you're trying to protect it from. And so when I suspected that, but I did not know that there were no animal studies to date on the SARS-2 coronavirus vaccine that's being tested right now in the state of Washington. This is a collaboration between the U.S. NIAID, that's Anthony Fauci, and Moderna. That's a company whose stock has skyrocketed because they went to phase 1 clinical trial with people. I suspected 2 weeks ago that there was no animal studies, and I looked around, and now it's been confirmed. And the media is talking about it. There has been no animal studies to show whether this is safe. And the reason why that's a huge problem in this case is because we already know from the SARS vaccine studies, the animal, not the SARS coronavirus 2, but the SARS coronavirus, that animals had increased risks of things like hepatitis, an increased risk of infiltrates. So I just want to read a few things here from some of the studies. So the immunized ferrets developed a more rapid and vigorous neutralizing antibody response than control animals, but they also exhibited a strong inflammatory response in liver tissue. That was for the SARS study. So there's another one. The VRPN vaccines, this is for SARS, not only failed to protect from homologous or heterologous challenge, but resulted in an enhanced immunopathology. Now, this term enhanced has been changed to enhanced immunity, and it's the wrong term. Peter Hotez calls it enhanced immunity, and because he uses that term, I came up with a new term. It's pathogenic priming. So it goes on. The vaccine resulted in enhanced immunopathology with eosinophilic infiltrates within the lungs of SARS-CoV challenged mice. They were sicker, and they had unacceptable high mortality rates. So we're looking at a knowledge base that has been thrown out the window. They know that it would lead to a vaccine being produced by them to be a problem if they did the animal studies, so they've skipped it. It's not as though they don't know about this history. So this pathogenic priming occurs almost certainly because there are the immunogenic parts of the protein in the spike protein in SARS and SARS coronavirus 2 match human proteins and cause autoimmunity on second exposure. That's what seems to be happening because these vaccines are adjuvanted.
So I just want to try and put that in lay terms for people watching, and please, if you can help me here, that would be great. So if we're getting a vaccine, we're starting to see inflammatory conditions, and just going back to what you said a minute ago, you're now more susceptible to other viruses. Is that
Nope.
a good way of framing it? Or
no, go ahead.
Okay, so help, yeah. So there's, we'll come back to that in a second, and of course, the autoimmune side of things is huge, right? So correct me if I'm wrong as well, but and perhaps this is maybe a little bit too further along here, and we can come back to it. It's my understanding that you mentioned earlier about Hubei province, right, and the vaccines and these trials. Do you think that that's part of why we're seeing such a huge number compared to anywhere else? Like, are they more susceptible? Is that what the vaccine is doing, or?
So it's interesting because pathogenic priming can occur no matter how you're exposed to it. There's a paper out by a science writer named Jason Tetro where he says that the coronavirus might actually be getting some help in terms of being more dangerous because there are other viruses that are similar to it that prime. This pathogenic priming, as I'm calling it, the pathogenic priming can occur in Hubei province if people are exposed to the spike protein or a relative of the spike protein through the processing and preparation and eating of consuming of meats in a market. But the question is, why now? As opposed to 100 years ago or 50 years ago. And what happened that was different is on January 1, December 1, 2019, China's new whole population vaccination program went into place. And so these people are walking around with adjuvant in them. Then they get exposed to it, just like the mice. That's the missing component. So whether it was a vaccine a secret trial, or whether it was just this mass vaccination program, and now they're exposed to the coronavirus, they're developing autoimmunity against tissues in their lung, in their liver, and kidneys. And so I have a paper under review right now that shows which specific parts of the proteins in SARS-CoV-2 are probably responsible for the immunopathology that we see, the pathogenic priming. And I hope that review process goes quickly because, this sounds all academic, and I don't want it to sound academic. I am so pissed off that they went ahead with a vaccine trial in the United States without doing the animal studies, that I want every person that within the sound of my voice to contact their congressmen and their senators and have them hold a hearing, an emergency hearing on immunopathology, right, of a pathogenic priming hearing on why they didn't do animal studies first, whose decision it was to go forward with the human trials, and why are they paying people $4,500 to take this vaccine? That is highly unethical. These people are unknowingly potentially trading a lifelong risk of more serious illness from coronavirus by being vaccinated. And so I have an article on this on LinkedIn. The article is kind of taking off on LinkedIn, which is good to see. But I'm asking the question, are Moderna and USAID poised to endanger the world population? Because if the United States gets a vaccine that's not animal safety tested, and it includes those unsafe epitopes, then the entire world could suffer massively in 2021, 2022, when another coronavirus outbreak comes. So it's the re-challenge with an infection that's the problem after you've been injected with this with an adjuvanted vaccine that has these proteins.
Right. And I think it's also important, I mean, at least from what I've seen, there's also no placebo controls at all either. This is happening in Seattle, where I think there's 45 people that they're wanting to test on, and no placebo, no animal safety studies. So, that's also quite concerning for me.
It should be concerning. So it's par for the course for vaccine safety studies to not use an appropriate placebo. I don't even think that they're using an adjuvant placebo. So if there's pathogenesis, that is disease causing by the vaccine from the biological components, only in the people that get just the biological component, then they wouldn't want to see that. So here's a case where it might be so dangerous that even a incorrectly placeboed vaccine safety trial might show that there's a problem. And again, the reason why we don't have a SARS vaccine is not because of anti-vaxxers. It's because vaccine safety science translational research worked, and it showed the world. And the authors of these papers, they actually say caution, extreme caution is warranted in bringing these vaccines into the human population. So please make a point sometime today to contact your congressmen and senators, write email, call the office, leave a message. We need thousands and thousands of phone calls about why there's no animal safety studies, and that we absolutely have to see that the very vaccine that they're using, not a similar vaccine, not something that's one-off, the exact vaccine that they're using in human beings has a safety profile that's okayed by animal studies.
Well, of course, what we're going to hear is, this is an emergency situation. We need to rush it to market. It would be unethical if we didn't, et cetera, et cetera. So I think that, the concern that I have as well is that given the hyped state that the media is creating, which I think to a certain degree is warranted, like we are facing a genuine pandemic. This is escalating rapidly. But I think what my concern is that people are just, without even thinking about it, without even looking into this, they're just going to roll up their sleeve and say, give it to me, and en masse. And again, the unintended consequences we're not sure of. But I want to add a couple of things here, James, and get, just to sort of look a little bit further ahead and where this is all going. Do you have any concerns over the virus itself mutating? So let's, for example, say we create this, COVID-19 vaccine, and next season or whenever it is, much like HIV, this thing just replicates and essentially alters its makeup, and therefore rendering the vaccine ineffective. Like, what are your thoughts on that?
So the specific vaccine that Moderna has is called an mRNA vaccine, which means that they can literally change it to any strain that's circulating very quickly, which is a benefit. I'll admit it. Mm-hmm. However, it still encodes the spike protein. And what we know from other studies is that a process called immune epitope suppression, and it's also called original antigenic sin, that if you're vaccinated against, say, this coronavirus this year and another one comes out, not only will the vaccine not be likely to work like the flu definitely mutates every year,
right?
But you could actually become more susceptible to a variant of the flu. Because your immune system B cells, it turns out, is confused about what it's doing. There's absolutely every reason to be concerned about mutations in viruses for which we have vaccines, including the measles. I was in Connecticut, and there was a Yale epidemiologist that testified to a committee that wanted to strip away the rights for people to say, I don't want a vaccine based on religious reasons, that the measles vaccine does not mutate. In fact, the measles virus doesn't mutate. The measles virus has something like 4.3 mutations, per replication. It's a huge. It's not as rapid as an RNA virus like the coronavirus. The coronavirus has a ridiculously high mutation rate, and also the very biology of how this thing works is if you're infected with two coronaviruses that are similar, they will recombine and create a hybrid virus. Okay, wow. So the recombination is essential to there's an alternative splicing-like mechanism where there's an S protein, a structural protein, and a spike protein, and it's called a polyprotein at the beginning. The polyprotein encodes multiple different proteins. And so there is variation. Because it's an RNA virus, the mutations that occur in the RNA, if you inject me with this coronavirus right now and I get sick and I get better in 5 days or 6 days or 2 weeks, whatever, and then you isolate the virus. By the way, this virus is known to be, you can shed the virus after you're infected for like 68 days in your feces. This is a big problem.
Wow. Okay.
Yeah, it's
That's
like
crazy.
Yeah. So
wow.
So I'm 68 days later, you isolate it. It's going to look very different. It's going to have hundreds and hundreds of mutations from just by being in me. This thing will adapt to my immune system, and if I pass it on to someone else, it'll adapt to their immune system. The net influx of mutations into a population determines the rate of evolution, and mRNA viruses have, among all the viruses, the highest rate of evolution because they have such a high mutation rate.
Wow. And then when you map that onto this not exponential, but you know what I mean, this rapid spread, obviously that just, accelerates the whole process. And now I've heard reports coming out of, this is all very recently, obviously, I mean, literally within the last couple of days, reports of people in China being reinfected after they have been cured. Any insight into that?
Yeah, absolutely. So it appears that you can be reinfected. There's multiple reports, not just in China, that I've seen now, that a person that had the infection and had symptoms then reacquired a new infection, and that's probably not unrelated to the fact that there's multiple strains circulating around, and our immune systems don't see it. The problem is, through this pathogenic priming, these people know that they have a secondary infection because they are even sicker the second time, right? So if you get if you get the virus one time and then you get the same strain, you're really not going to notice it. The immune system should be able to take care of it, right? But if you get a slightly different one, then the immune system might not really know how to fight that different one off. My study that's under review also shows that this particular virus, of the immunogenic epitopes that match human proteins that could cause autoimmunity that I was talking about, a third of them, right, 7 out of 21, I think it is, have matches or homology to very key players in our immune system. So the actual pathogenesis of this virus may involve an attack, our own body attacking our immune system cells, our immune system proteins. And if that's the case, then people that have mutations through no fault of their own, just through the accident of how they were born, they have genetic mutations that make their protein one step closer or more similar, to the viral protein. Those are at highest risk, and nobody knows who they are because they're not doing individualized medicine when it comes to, immunity. We have the ability to sequence our genomes, right? If somebody gave me their whole genome sequence, and I'm not asking to do this, I'm not offering a service, and I won't analyze anybody's data, but if theoretically, if somebody gave me their whole human genome sequence or their exome sequence, I could analyze it. I could tell them this virus is likely to, if it induces autoimmunity, it'll attack you here, and it'll attack you there. I can tell you the protein, I can tell you the tissue. Why? Because it only takes a small segment of the epitope to match to confuse the immune system. So if we go back to mutations for a minute, it's understood in viral evolution that if you are infected with a virus in a population, it spreads throughout a population, it's going to kill off that variant that comes in that's really lethal. It's really lethal. It'll kill off individuals that are most susceptible, probably through genetics, but it'll also, with those people dying, it'll take the virus, takes itself out of the running. So the strains that are left are going to be less virulent. The strains that are left are not going to be less transmissible. They're going to be more transmissible. The ones that survive are more transmissible. So there's this, it's called an adaptive landscape of transmissibility versus virulence. I wrote about this on my blog many moons ago now about Ebola. The transmissibility is the ability of the virus to actually transmit. That's supposed to be expected to get better and better and better and better for the virus. So it's more likely to get it, but the virulence is supposed to go down further and further and further. So if we the interesting thing is, if we interrupt that process, and it's a conundrum at this point, if we interrupt that process with a vaccine, we've changed the adaptive landscape in ways we don't understand. Yeah. And people will say, well, we're playing God, we're messing with nature. I'm just talking as a scientist. When you have a new virus and it has such strong propensity, I believe, on the basis of the studies that I've done, to induce autoimmunity, are we making it more transmissible? Are we making it more lethal? There's in an adaptive landscape where you have this third factor, it becomes a three-dimensional landscape. It's not just a two-dimensional landscape. And that three-dimensional landscape, there are adaptive peaks. The virus fitness is what I'm talking about. The adaptive peaks on the human landscape is here without the vaccine, but the adaptive peak might be over here with the vaccine, and the virus will go towards whatever that adaptive peak is, and we don't know what that is. That's the problem. There are too many variables.
Yeah. So I'm going to just to bring us into the close here. Obviously we're going on about an hour now. You talk about having such close similarities to the human tissue, et cetera, et cetera. Not tissue. I'm botching the nomenclature here.
That's fine, because proteins are part of tissue. It's fine.
Right. But there's talk out there. You and I spoke about this before we hopped on here. In the early stages, and given the origins of the virus, given the fact that it's coming out of a very, I think it's a level 4 security lab, one of the few in the world that deals with highly infectious pathogens. Do how did this virus come about? Like, do you think it was a natural mutation of something that existed already? Or do you feel that this was somehow engineered for research purposes, for whatever purposes? Like, do you have any thoughts on that? And again, I'm sort of putting my tinfoil hat on a little bit here, and I don't want to put you on the spot, but if you have any insight into that, I know people are asking that question.
Okay, so I think we can rule out biological weapon. The reason why I think we can rule out biological weapon is because looking at the sequences that I've looked at, there doesn't seem to be any credible evidence that there are new elements here that necessarily could only have come from, somebody with a pipette in a lab doing, transfection experiments and using pea shuttle and so on. But it doesn't mean that it didn't come from a lab. There's no evidence that it wasn't a natural virus that was being studied in the Wuhan Institute for Virology, or by the military of China for that matter, and accidentally escaped. And I'm not just trying to play peacemaker here. I really am basing this on data. I've looked at the data where I did an in-depth analysis, another IPAC study that's under review right now, where I looked at the functional motifs of the spike protein to see if there's anything different about this spike protein compared to the SARS coronavirus. And I noticed that there is a short, what's called, and for this is, I speak technically. People say, could you dumb it down? You're using too many big words. I speak technically because there are scientists that watch my videos as well.
Exactly.
But there's a short N-terminal segment compared to the SARS vaccine in the SARS virus in the spike protein. There's two missing middle segments of functional motifs. The amino acids are there, the sequence is there, but there's two that are missing. The SHE3 is missing and the KXD is missing. And then at the end, and this is going to blow things up again here, but there's a GP40-like motif at the end. Now, GP40 is the type of an envelope protein that we see in like an HIV. But evolution can put that kind of thing there too if it is adaptive for this virus to attack immune systems for some reason. See, so it's called convergent evolution. So what's the most likely scenario? Just knowing that, we don't know. But if you look at the time frame I asked the following question of the national databases, of protein databases. Given that we know this motif pattern, a short N-terminal, two missing in the middle, and a GP40-like, given that we know that could be a signature, is it present in any of the other beta coronaviruses? And the answer is no. There's a bit of it up in one, the short N-terminal, but the whole pattern is not there. And then I asked the following question. All right, so if it's not in the other beta coronaviruses, it looks like it's brand new. What's the earliest date that anyone published a sequence that had this? And I found it in a SARS, what must be a mislabeled SARS coronavirus. The only SARS coronavirus that had it was a sequence that was published in 2005 by Chinese researchers who had isolated the virus from bats in Hong Kong. Okay? They in the spirit of transparency and to help humanity understand coronaviruses and SARS, the Chinese published this sequence, and they didn't know that it had this spike protein-like structure. It has the SARS-CoV-2 structure, the structural motif pattern. So they were doing the right thing. They were being good citizens, and they published it. Our scientist at the University of North Carolina, Dr. Barrick's lab, downloaded those sequences. One of them, it downloaded 4 sequences. One of them was one of these sequences. And I know that Barrick and his team didn't know that this was a SARS coronavirus, like SARS-2-like virus, or that it was even something different, because they calculated what's called a consensus sequence among all 4 of what they thought were SARS. It's just an accident of history that I noted that, well, it's right. I asked it because the right question, because I'm a scientist, but it was just an accident of history that the history of these viruses included a SARS-CoV-2-like signature way back in 2005. Our scientists were messing around with it in 2008. Had they only downloaded one sequence, and had they only downloaded this one sequence that the Chinese published, they would have reconstituted the SARS-CoV-2-like virus in the lab in 2008 in the United States, and the outbreak could have started here. But they didn't. They calculated a consensus, so they got lucky. And by the way, this is so scientifically important because there are a lot of gain-of-function studies, which we're all suspicious of. Why would you want to give a virus more functions? Well, sometimes we give gain-of-function studies so that we can allow the virus to infect, say, mouse tissue, so we can study the virus in mice. If it can't infect the mice, then we need to make it be able to do that. But there may be studies of compounds that are effective at treating this. If people go and look at the actual protein motif signature, we'll look at the spike protein of the viruses that you've been studying, the SARS viruses, and see if you are not also accidentally studying treatments for SARS-CoV-2 and you didn't know it. And that, right? Huh. So imagine you have two viruses, but you don't know it. You have the SARS and the SARS-CoV-2, and it was already there in 2005. Why? Because it's been around for a while, and they just sampled from nature. And you're testing out a compound, and you say, well, okay, so I'm going to test it on this mouse, I'm going to test it on that mouse, and test it on that mouse. Well, if the third mouse was infected with SARS-CoV-2 and you didn't know it, then you have a heterogeneous population, it's called, and the properties of the treatment we might have more effective treatments than we know, because we really don't understand what we're doing when we're looking at the functional differences among viruses. So there's plenty of slop, I would say. And I prefer slop and profit motive over bioweapons and conspiracy, 100%.
Yeah.
Right?
Okay.
Because we are not as smart as we think we are as a species. Scientists think that they're really super smart. They have this Dunning-Kruger effect that, medical professionals as well. They're actually taught, you're going to make mistakes and kill people, but that's okay because you're a god, right? You don't want to think that way. You want to be humble. And that's why when I first came out and I said that I think that this spike protein was related to P shuttle SN, I've spent the last month, instead of working on blogs about therapeutics, I spent the last month talking about this issue that no, it was just a hypothesis. No. There's three other hypotheses. I've subsequently tested the hypothesis with phylogenetics, and that's how I found the 2008, the 2005 sequence. I believe that scientists are exonerated this time, but it does not mean it did not escape from the Wuhan Institute for Virology lab. That's a different question. If they're brought in one to find out what it's like, they didn't modify it, and it escaped, and it still had these properties, okay. If they brought it in and they modified it for gain of function, and it escaped, okay. The Chinese diplomat right now just made a very, very cursed tweet. Last night, I think it was, the Trump administration responded yesterday, the Trump administration responded and said, no, we know where it came from. The Chinese diplomat implied that somehow the U.S. Army may have brought this virus into Wuhan and set off this fire, and that our scientists need to be more transparent, that they're giving back to us the treatment that they think that they're getting from me, frankly, because I'm in touch. I can't tell you how many Chinese national scientists have contacted me. They've actually written a paper, in a peer-reviewed paper, saying that they were appalled that I would suggest that this could possibly come from a lab. And all they did was a simple multiple alignment of the P shuttle compared to other proteins. That doesn't prove it didn't come from a lab. What that proves is that the missing segment that the original Chinese publication could not identify, that missing segment, and then another study by a Greek team, they could not identify it. They called the middle fragment, has a spike protein part to it. That's all it proves. It doesn't prove where it came from. So my study, which is under peer review, demonstrates that they're exonerated in terms of artificial origin. We know it came from nature. That's what I think. Okay. And we don't know that natural sequence didn't escape from a Chinese laboratory. And the fact is, between 2003 and 2014, there were 6 escapes of SARS from a laboratory. 6 of them.
Okay.
And 4 of those came from a lab in Beijing. So sloppy human beings messing around with viruses in the lab with a profit motive is obviously a recipe for disaster. And let's not kid ourselves. China is a communist state, but there is a profit motive in terms of getting involved and beating people to the market for a vaccine. There's no doubt about it.
Yeah.
We need to knock that off. We need to make vaccines so unprofitable. We should nationalize it and say, no, you cannot make a profit. You have to be a not-for-profit. If you turn a profit, you're unethical. We need to go that route with vaccines. And everything's on the line for them, too, because they're absolutely, the truth is coming forward that they tried to suppress since 1986 or before. The truth is coming forward about real risks and attendant risks of vaccinations, and everything is on the line for them, which is going to make so many people in our immediate community so suspicious that this is just a way to shut down our discussions and this kind of thing. Listen, I told you earlier what we think the real risks are. If you have an elderly person, take care of your family. Make sure that they don't get sick. Don't, God forbid that they should get this thing twice, right? Given everything that we've talked about. All right? And I'm leading the fight against this rush to human trials with vaccines that are not inadequately safety tested. They have shown all of their cards. All their cards are on the table. And all we have to do is alert our politicians to that. You know what? They have set us up for a fight when it comes to our very lives. And they have to demonstrate that this thing is safe in animals. They could do that study anytime. They don't have, it's not too late. They could do that study anytime. Yeah. They could do it tomorrow. So we need that study done. We need it done with a large number of animals. We need it done with all the vaccines for SARS, coronavirus 2. And it's not that I don't care. It's clear that very clearly care about people that are going to get sick and die from this. I've been like you said at the beginning of the segment. I was one of the first early voices saying we got to shut this down. I'm one of the loudest voices saying we got to protect the medical infrastructure, right? It's going to be a disaster worse than Italy. And yet you still have to go by the book in terms of translational research. If you don't, you're going to be potentially putting the entire world at risk of 20% mortality across the board next time it comes around.
Well, which would obviously be totally catastrophic. So, James, I think, let's wrap it up there. Thank you so much for taking time out to speak with me today. And, I think, I hope anyway, for those of you watching out there and listening to this, that, I know some of the terms and some of the way that James presents things. He is a top-notch scientist. So we're trying to appease the scientific community while at the same time trying to sort of bring people who are not scientists into the fold so that we can sort of make sense of what's going on. So it seems to me, James, like the best thing that we can be doing right now is really just to be isolating ourselves, limiting human-to-human contact, taking herbs and therapeutics, antivirals that we know may work and that don't have negative side effects, and just sort of see how this whole thing plays out.
Yeah, absolutely. So if you've taken the time to stock up your pantry and you have a deep pantry for a couple of weeks, 2 or 3 or 4 weeks, if you've done that, right, then it's time to take care of other people. It's the most natural human thing to then go, how can I help people that I know and love? How can I help my neighbor? And the best thing that you can do, and I'm going to say this point blank, the best thing that you can do to help your neighbor, your children, your grandparents, is to call your congressman and call your senator. The most important threat that we have right now is not this immediate virus. It's the outcome after they vaccinate everyone. And the people still get the virus, or people get a related virus. If you got to think long term in this, but certainly, share the advice of getting your pantry ready. Understand that you are part of your government in the United States. You have the right and the responsibility to participate in your own government. Don't just throw your hands in the air and say they're all corrupt, because they too have children. They too are susceptible. They too will be vaccinated. Okay, people are. I can hear people that I know, the specific people that I know and love right now, that say, no, they won't. They won't take the vaccine. They'll put it on all of us. No, they will take this vaccine to build confidence in the rest of the world. They'll line up all the congressmen and senators in a meeting, and they'll vaccinate them. There'll be a big show. It'll only be sailing. Okay, well, we could do this all day long, but what are we going to do? Stop listening to our podcast and go call your congressman and go call your senator and demand animal safety studies for every SARS-CoV-2 vaccine and for every vaccine going forward. And thank you so much, Brett, for hosting this. I really appreciate it.
Yeah, and likewise, James. I mean, for those of you tuning in on Facebook, wherever, obviously this is a total renegade meeting today. We literally just came about this last night. So thanks everyone for tuning in. And, I just wish everyone nothing but the best. And I think that, my sort of parting words here is, let's really try and keep the panic down. I don't think panic is going to really solve anything. I think just being vigilant, self-quarantining, staying in isolation, eating right, getting rest, taking whatever you can to support your immune system, help with antivirals and so forth. I think that's really the way to go here. So thanks, James. I think we're going to wrap it up, and I'm going to catch up. I know we're doing a Facebook Live feed here, so we'll see if there's any comments there. And have an awesome day, everyone. Thank you.